Hey guys, this is Kendall Terry, and this is Cellular Respiration, a harvesting of chemical energy. In other words, it's the making of ATP. Very important for the cell. So why is respiration important? Let's start very basic. You've got the consumption of food and oxygen to produce carbon dioxide and water, and energy. And energy is going to come in the form of ATP and heat. So you can see here the chemical equation for cellular respiration: C6H12O6 plus 6CO2 plus 6O2 oxygen is going to make or yield in carbon dioxide, 6 carbon dioxide molecules plus 6 water molecules plus energy in that form of ATP and heat. You can see it in the graphic illustration here. You've got light energy, of course, comes into photosynthesis and produces organic molecules. Specifically, the one that we look at most of the time is glucose. Glucose and oxygen go into the mitochondria and produce ATP and heat energy. And your body produces tons of heat energy on a regular basis. It's how you stay 98.6 degrees, but you also have excess heat that you're letting off all the time. And then that process of cellular respiration in the mitochondria is going to produce carbon dioxide and water, which is what photosynthesis will use in the presence of light to make glucose and oxygen. So it's just a circular pattern that we're going to discuss throughout this section. So looking still more at why is respiration important, it's important because it's an exergonic reaction. It releases energy. And that's important because as you eat the food you eat them for energy. So if this process didn't release energy, then it wouldn't actually give you any benefit. But because this reaction is exergonic, meaning it's releasing energy, then that energy can be harvested. And that's the whole goal of cellular respiration, is to harvest that energy. As that glucose molecule that you eat comes in and we break it apart, that releases energy, and that release of energy has to be picked up and turned into something your cells can use, and ATP is what it's going to be turned into. We're going to look at that mechanism of picking up that energy as that happens. All food that you eat can be metabolized as fuel. That includes the carbs, the proteins, and the fats, which are the three most important things that you eat that your body's going to use for energy. These reactions are what we call redox reactions. Now, depending on what you've had so far in school, you may or may not have ever heard of redox reactions. Redox reactions are reactions that gain and lose electrons. And the things that are being involved in this process are either being reduced through a process called reduction, or oxidized through a process called oxidation. Now, the way to remember what goes on with each is this little statement here: Leo says Ger. Leo says Ger stands for loss of electrons is oxidation. So if a molecule is losing electrons, it's being oxidized. Gain of electrons is reduction. So if it's gaining an electron, then it's being reduced. So when we look at the chemical equation for cellular respiration, you can see that here. Glucose is being broken down into the carbon dioxide molecules. So to do that, it's having to lose electrons. So it's being oxidized, whereas oxygen is being turned into water, and it does so by gaining the electrons that glucose lost, and that means it's being reduced. So when we look at cellular respiration equation, it gives us an idea really of what we should focus on in cellular respiration, which is the electrons. This whole process, I would tell you that if you want to know what's going on, if you can tell me where the electrons are, then you have a good idea of what's happening in cellular respiration. So what is being oxidized? What is losing electrons at any given point? What is being reduced? What's gaining electrons at any given point? Until you go from glucose to water, then you know the process is done. So there's three main steps of respiration. There's glycolysis, citric acid cycle, or the Krebs cycle, and then oxidative phosphorylation, which actually involves two processes: the electron transport chain and chemiosmosis, and we'll talk about those in a minute. So here's an overview of the process. You've got glycolysis that takes place out here in what we call the cytosol or cytoplasm of the cell. So that is where glucose is going to enter. As glucose enters glycolysis, it's going to be turned into pyruvate. That's also going to be a release of ATP through substrate level phosphorylation. We'll talk about that in just a minute. Pyruvate will go into the mitochondria, and during that process of going into the mitochondria will be turned into acetyl-CoA. That'll go into the citric acid cycle, and the products of the citric acid cycle will enter oxidative phosphorylation and eventually get your major output of ATPs through oxidative phosphorylation there at the end. So let's look at each step individually. Glycolysis is splitting of glucose in the cytosol or cytoplasm. It's 10 steps and no oxygen is needed. This can be referred to as an anaerobic process. Anaerobic because oxygen is not needed for this part of cellular respiration to take place. Here's the kind of breakdown of it. You do not need to know all 10 steps for the scope and sequence of this course, but you do need to know that glucose comes in. Glucose has to get, this process has to get going somehow, and you can think of this as kind of like activation energy or whatever, but there has to be something to kind of kick this process off. So what happens is 2 ATPs will be added. As 2 ATPs are added to the glucose, that gets the ball rolling, and through substrate level phosphorylation, you make 4 ATPs and 2 NADH molecules and then 2 pyruvates. NADH molecules are referred to as electron carriers, and I think of them as dump trucks. They carry electrons from one process and dump them onto another, and we'll look at that in more detail later. But right now you need to know glycolysis takes glucose in, you use 2 ATPs to get the ball rolling, it makes 4 ATPs, so that gives you a net gain of 2, and then it makes NADH and pyruvate. It makes 2 pyruvate for every 1 glucose. So how is ATP made? What is substrate level phosphorylation? Well, that is a process that ATPs are produced from transfer of phosphate group from a substrate to an ADP. So here you've got an enzyme, you've got the substrate, you've got an ADP molecule. So the enzyme is going to take that phosphate from the substrate and attach it to the ADP, making ATP. And this is the process of making ATP that takes place all the way up into chemiosmosis and electron transport chain that we'll talk about later on. So how do electrons get from glucose to oxygen? Well, they use those electron carriers. Every time we break glucose, that bond that's holding that molecule together is an electron in essence. That's how we describe bonds, as electrons. So when we break bonds, we release electrons. Those electrons have to be picked up by somebody to get to oxygen. So electron carriers do so. Now NAD plus, nicotinamide adenine dinucleotide, is the dump truck that it uses mostly in this process. We'll talk about another dump truck later on. This is a coenzyme. It accepts 2 electrons and a hydrogen, and it turns into NADH. Let's look at that molecule. So here is NAD+. The white part up here is the part that will change. So whenever it accepts its 2 electrons and it is reduced, remember, accepting electrons, that's reduction. When it accepts the electrons, it accepts a hydrogen with it, and it turns into NADH because of the extra hydrogen gain. It will eventually dump these electrons. That would be losing electrons, so that process is oxidation. Remember, we're always wanting to know, are you gaining electrons or losing electrons in this process? So how does pyruvate then get into the mitochondria? So far, everything we've talked about has been outside the mitochondria. What it does so through active transport across that double membrane of the mitochondria. You'll remember the mitochondria has a double membrane. That inside one folding back and forth on itself, that'll be important later on. There's a 3-step process. So here we've got pyruvate out here. As it's brought through active transport into the mitochondria, it will lose a carbon dioxide. Now remember, if you're breaking carbon dioxide off, you're breaking a bond, so you have to release an electron. So notice, here's a dump truck picking up an electron, NADH, and it's going to attach a coenzyme called coenzyme A to it, and it turns into what we call acetyl-CoA. All right, so what happens during the citric acid cycle? So now we've got acetyl-CoA in the mitochondria, but something still has to happen because we haven't completely broken it down yet. There's still carbons attached to each other. We want to break all of those apart. This happens in the mitochondrial matrix, meaning the very inside part of the mitochondria. There's 8 steps. You do not need to know each of one of those steps for the sequence of this course, but it has to spin twice, 2 times, for every glucose, because remember, one glucose made 2 pyruvate. Those 2 pyruvate each made acetyl-CoA. So you've got 2 acetyl-CoAs that are sitting there now in the mitochondrial matrix. Each of them have to be broken down, so the Krebs cycle has to turn 2 whole times to break that down. So we look here, pyruvate, of course, out here is turned into acetyl-CoA. Acetyl-CoA goes into the citric acid cycle or the Krebs cycle. As it turns, it's going to release 2 carbon dioxide, so it's breaking a carbon dioxide off. If we break a carbon dioxide, what do we release? We release energy in the form of electrons. NADH is there to accept those. FADH2 comes into play. That is also an electron carrier, and it's just carrying electrons. It's just a different dump truck company, but it's still doing the same job. And then you also have some ATPs that are made through substrate level phosphorylation that we talked about earlier. So how many ATPs have we made so far? We made 4 total, 2 in glycolysis and 2 from the Krebs cycle. So we have a total right now of 4 ATPs, total net gain ATPs that have been made. All of them have been made from substrate level phosphorylation. That's not very many ATPs. There's got to be more to this process. So what do we have? We've got ATPs, but then we've also got all of our electrons right now are stuffed into electron carriers. We've got 10 NADHs out there, and we've got 2 FADH2s that have all been made from breaking glucose all the way down. So those electrons have to still go somewhere. They're in dump trucks. Dump trucks are never the final place that anything goes. That's just the transport. So what we're going to discuss in the next lecture is what happens from there.